and welcome to our webinar. My name is Kevin Knopp, co-founder and CEO of 908 Devices. Boris and I are excited to share with you a new product we've been working on that we think all of you will find exciting too. We view the bioprocessing market as being at an inflection point. The pipeline of therapeutics is growing and increasing in complexity. As the industry progresses on from small molecule drug to large molecules and proteins to ever more complex advanced therapies, the demands on process development and manufacturing have never been greater. The industry is embracing the concept coined Bioprocessing 4.0 to lower errors, the cost of goods, to get better data, and for more efficiency. To enable this, there's a need for more analytics, more real-time control, more digitalization overall, and more prediction. Continued advances in process intensification, the need to improve efficacy, and the regulatory push to implement process analytical technology, PAT, call for new and improved methods for directly monitoring and controlling key process parameters, KPPs, and critical quality attributes, CQAs, to achieve maximum yields, improve product efficacy and quality. To this end, 908 Devices first introduced our ZipChip device for offline CQA analysis as an alternate to more complicated and legacy liquid chromatography, LC, technology. ZipChip is a microfluidic capillary electrophoresis, CE, technique that has been shown to be 20x faster than HPLC for multi-attribute method, MAM, analysis that requires no or little method development. Next, we introduced a first-of-its-kind device called Rebel that is a small desktop form factor, combining both our mass spec technology and our CE and machine learning technology to serve as a tool for process development to quickly and easily measure fresh and spent cell culture media at line, right next to the bioreactor in the PD lab. In just about 7 to 10 minutes, quantitative concentrations of a complete panel of 32 analytes, including amino acids, can be obtained. ZipChip offline and Rebel at line are great, very impactful, but we are an innovation technology company, and if you look forward, limited process insights can be gained from manual daily sampling. Hence, easy to implement continuous monitoring of nutrients and metabolites are needed to enable dynamic feedback control. The key enabler to create an ecosystem of connected online analytics is the aseptic interface. The aseptic interface must, 1, maintain sterility, most critical to not contaminate the culture with foreign bacteria. 2, use minimal or no volume. These are precious samples, not to be wasted. And 3, be cell-free and flexible, to take only what is needed, as glucose, lactate, amino acids, and other key process parameters don't need cells or even proteins. That is what gets Boris and I excited to be speaking with you today. We've been working to address these challenges to enable online analytics, and we would like to introduce you to our newest and first ever online device offering from all of us at 908, MAVEN. MAVEN is an online GMP-ready glucose and lactate analyzer that can not only measure, but can also control. To tell you much more about why glucose and lactate monitoring is so important and much more about MAVEN, I'd like to hand it over to my colleague, Boris.
Thank you, Kevin. Hello, my name is Boris Aleynik. I'm a director of life science product marketing at 908 Devices. In the rest of this webinar, we will review the importance of continuous monitoring and controlling of glucose and lactate in cell culture and fermentation, followed by the introduction of our latest product called MAVEN. But first, let's start with the basics. A lot of upstream process variability can naturally be attributed to the metabolism of cells. After all, cells are the factory or the actual product themselves. Fed batch is the go-to process for production of most biologics. As we all know, one challenge of fed batch is that cell metabolism changes over the duration of culture. For the most part, this is due to changes in available nutrients and accumulation of metabolites, which produce those distinct phases of cell growth, namely the lag, the exponential growth, stationary, and the decline phase. Glucose, being the main source of energy, is a critical parameter to monitor and control in most cell culture and fermentation processes. During the exponential growth phase, up to 70% of glucose is oxidized via glycolysis into pyruvate. Large portion of pyruvate is then converted into lactate, while the rest supplies the TCA cycle. High concentration of lactate acidifies the medium, and to compensate, base is added. This increases osmolality, which in turn can inhibit further growth and can lower product yield, as well as negatively impact certain product quality attributes. It has been shown that a shift in metabolism from lactate production to net lactate consumption is associated with high-performing process. While being highly desirable, the mechanism behind the switch is not fully understood or easily controlled. So how much glucose and lactate cells actually need to be happy? Glucose and lactate are closely linked and are influenced by other nutrients and metabolites, such as the amino acids. Even a small difference in timing of feeding may cause cultures with otherwise very similar metabolic kinetics to diverge to different outcomes. Optimal concentration of glucose and lactate, which should be maintained, depend on several factors: type of cells being cultivated, processing strategy, physical environment, and many other factors. But one thing is clear: they need to be just right and optimized on case-by-case basis. Too much glucose, for example, can quickly induce high levels of lactate and negatively impact on productivity and product quality. But too little of one or the other isn't good either. Finding the right level and feeding the right amount at the right time is key. There are a few feeding strategies that are available. We can feed at specific intervals with constant volume, what we call the bolus feeding, or we can feed based on a predetermined culture profile. A more advanced strategy would be to feed continuously based on feedback from measured parameters related to cell growth and metabolism. Bolus feeding tends to cause fluctuations in available glucose. These fluctuations deregulate metabolism and may inhibit viability and growth. Often in fed batch process, we try to increase protein expression by increasing the amount of concentrated feed. But this, as I was pointing out earlier, will likely cause increased accumulation of toxic metabolites, which will destabilize the process and will have the opposite effects on protein expression. Not to mention, this can have negative effects on certain quality attributes of the product, such as the profile of glycosylation. Dynamic glucose feeding, based on real-time measurements, can help achieve better overall process performance, including prolonged viability, higher viable cell density, lower lactate concentration, and improved quality attributes. The potential benefit of controlling feeding to improve process robustness and product quality are well documented in scientific literature and go back many years. There are many peer-reviewed publications that demonstrate the influence of glucose and lactate on health of cells, and consequently monoclonal antibody titers, CAR T-cell therapy efficacy and safety, and viral vector titer. For example, back in 2011, Gagnon and team invented glucose-restricted fed batch process, what they called high-end pH-controlled delivery of glucose, HIPDOG for short. This feeding strategy utilized pH signal for controlling glucose feed and demonstrated reduction in lactate and doubling of final titer in CHO cell lines. A study published by Sukumar and team demonstrated that inhibition of glucose uptake can enhance anti-tumor activity of T cells. When CAR T cells are injected back into the patient, they compete for nutrients with cancer cells, hence a lot of interest in methods for adaptation of T cells during manufacturing of cell therapies. Another study by Fan and co-authors demonstrated that amino acids and glucose metabolism affects glycosylation of monoclonal antibodies produced in fed batch CHO cultures. And more recently, Kumar and colleagues showed that control of glucose supplementation increased viral titer in production of adeno-associated viral vectors. Regardless of application, robust real-time monitoring technologies are needed in order to enable implementation of feed control strategies. Today, the industry relies on a combination of multiple methods for monitoring and controlling of process and product quality. Back in 2004, FDA in the PAT guidance had provided the terminology, what we often hear today when talking about different measurement methods: the offline, at-line, online, and in-line. I'm sure most of you are familiar with these methods, so I will not go through them in detail, but it's worth noting that manual sampling required for offline and at-line analysis, while necessary for some parameters, has multiple inherent drawbacks. It is invasive and carries the risk of process and sample contamination, consumes bioreactor material, which is a significant issue when working in small volumes. Even when SOPs are followed to the letter, there is still potential for operator-to-operator variability. It is labor-intensive and requires operators being present during off hours. Additionally, the low frequency of sampling and the lack of real-time data makes it impossible to react to changes in the process as they occur. Because of these drawbacks and the inability to control the process in real time, there is a lot of interest in the industry in online and inline technologies for real-time monitoring and controlling of critical process parameters. So let's meet MAVEN, a process monitoring and feeding control device for cell culture and fermentation processes. MAVEN offers continuous monitoring of glucose and lactate simultaneously, as often as every 2 minutes. The low limit of detection for glucose is 0.01 grams per liter and 0.5 grams per liter for lactate. This enables accurate control of concentrations, even at very low levels, which is highly important in certain applications such as cells and gene therapy. Measurements are performed without loss of bioreactor volume and no additional risk of process contamination. We designed MAVEN to support all phases of drug development and manufacturing, so it is GMP ready. It can be used with majority of bioreactor and fermenter types and sizes. Finally, it is easy to operate, set up and maintain, and has a small footprint. And this is where our technology makes its mark. MAVEN makes it possible to monitor glucose and lactate continuously online. This enables dynamic control of feeding to maintain just the right levels of glucose and lactate to enhance process performance and ultimately more consistent quality of the drug. In fact, inline monitoring and real-time released Bioform Working Group ranked real-time monitoring of glucose as high priority due to the potential increase in product yield and gain better control of glycoforms. So let's look at how MAVEN actually works. At the heart of MAVEN is the selective flow diffusion technology and enzyme-based unparametric biosensing. An autoclavable or single-use probe is inserted into the bioreactor or integrated with single-use bioreactor bag. Alternatively, a flow cell can be used to monitor a spent media stream or if there's a lack of space for insertable probe. At the beginning of the measurement, clean buffer solution is pumped from the buffer bag into the probe, and the buffer flow is paused. The probe is a semi-permeable membrane that separates the buffer from the bioreactor environment. The cutoff rating of the membrane limits the molecular weight of things that can pass through the membrane. Glucose and lactate molecules are quite small, less than 180 daltons, so they can diffuse into the buffer solution, but larger molecules and cells are retained in the bioreactor. Hence, there's no bulk sampling and no loss of volume. Once glucose and lactate have time to load into the buffer flow is resumed, and the loaded buffer is sent to the measuring cell. The concentration of glucose and lactate is determined by a highly selective enzymatic biosensor, which generates electrical signal proportional to the concentration of analytes in the process. The signal is used to control the feeding pump. Lastly, the used buffer is disposed of into the waste container. And this cycle repeats as often as every 2 minutes and enables the dynamic control of feeding. So to recap, diffusion flow analysis with measurement by enzymatic biosensor provides the real-time measurement of glucose and lactate without loss of volume or increased risk of process contamination. MAVEN is designed for use in process development labs and meets the requirements for PAT, making the transfer to GMP quite straightforward. And it actually works. For example, as we see on the left graph, the biosensor is quite stable, so there's minimal drift over the operating life of around 5,000 measurements or 30 days. For longer run times, biosensor can be easily replaced without interrupting bioreactor operations. On the right graph, we see that measurement is very consistent and linear over the wide concentration range. Overall, MAVEN meets the important characteristics for online analytical methods. It is precise, stable, linear, has great selectivity to glucose and lactate, provides fast response time necessary for real-time control of feeding, maintains process sterility, requires little maintenance, is GMP ready, and is easy to operate and integrate with bioprocess systems and automation. 908 Devices is involved in an exciting collaboration to integrate MAVEN into the CHO process and implement dynamic glucose feeding based on real-time measurements of glucose and lactate. In this study, a monoclonal antibody expressing GS CHO cells were cultured in a 10-liter bioreactor in the fed batch process. Lactate and glucose were monitored online with MAVEN. Feeding pump was controlled via MAVEN's integrated PID controller. Glucose was maintained very close of 2 grams per liter set point, and overall, the culture showed a favorable lactate profile with a clear switch to lactate consumption. On the left-hand side, we see the plot of online glucose and lactate measurements collected every 20 minutes. Glucose rose to 5.6 grams per liter on the day 7 due to a temporary issue with the feeding pump, but after it settled very close to the 2 grams per liter set point. The yellow markers indicate the instances of supplementation with efficient PET-C+ and correlate with the spikes in glucose concentration. The green areas at the bottom show when glucose feeding pump was activated by the PID controller using the online glucose feedback signal from MAVEN. Lactate, the orange points on the graph, presented a typical switch profile in production to consumption. On the right-hand chart, we see what viability was at 60% at the end of the run, and viable cell density peaked around 22.5 million cells per milliliter. So this project is still ongoing. In this study, published in 2015, a group at Biogen developed an enhanced process to increase productivity and robustness in HEC293 cell line, producing recombinant protein. This publication described the use of online monitoring in combination with logic control that simultaneously monitor and control glucose and lactate, resulting in cell culture process improvements. They observed significant decrease in lactate production, were able to prolong culture viability, and significantly increase titer. And all this was done by closely monitoring glucose and lactate and maintaining glucose at relatively low levels of 0.2 to 0.5 grams per liter. We can see on the left graph what lactate concentration was reduced by maintaining low glucose conditions with automatic control of feeding. This controlled process also showed increased longevity and higher titer, as you can see on the right. So let's talk about how you would use MAVEN. We know what glucose and lactate are key parameters in majority of cell culture and fermentation processes, whether it's manufacturing of monoclonal antibodies with CHO cells, CAR T cell or stem cell therapies, production of viral vectors, or plasmids in E. coli fermentation. We designed MAVEN to support a variety of bioreactor types and sizes, from benchtop systems to GMP clinical and commercial manufacturing. Autoclavable, steam sterilizable, or gamma sterilizable single-use probes and flow cells are available to help integrate MAVEN with a variety of bioreactor fermenter types. And when it comes to integrating MAVEN with process control, automation, and data acquisition systems, there are multiple communication options, including the analog, serial, OPC UA, and Modbus TCP. So before we conclude, I'd like to summarize what MAVEN brings to the table. It offers continuous monitoring of glucose and lactate to improve process understanding and optimization with up to 30 measurements per hour, which translates to 720 data points per day for each analyte. It can help maintain tight control of glucose concentration even at very low levels, which is especially beneficial in cell therapies. MAVEN enables development and implementation of dynamic substrate feeding strategies. It allows to reap the benefits of continuous monitoring without loss of bioreactor volume, which is especially important when working in small volumes. There are many types of bioreactors out there, and we made sure that MAVEN is flexible enough to integrate with majority of them. Finally, we designed with ease of use in mind to help process development scientists focus on bigger picture, rather than babysitting their bioreactors, performing manual sampling and offline analysis, especially during off hours or when access to the lab may be limited.